Okay, hi guys. For the genetic linkage problem set, I will be doing question 1 with you guys because I feel like this particular problem set is a little bit tricky. And it would be better if you guys tried it out on your own and then watch this video, but you could also watch this video as you are doing it. Okay, so let's tackle question 1. It says you're doing a genetic experiment with the fruit fly, and in the P generation, which is the parent generation, you cross 2 true breeding flies. Remember, true breeding always means homozygous. The female parent is brown and wingless. The male parent is black with normal wings. And all the flies in the F1 generation are brown and have normal wings. So if we cross 2 homozygous organisms, all of the F1s are going to be heterozygous. Well, if the 2 parents have different traits, then all of the F1 generation are going to be heterozygous, right? So if you crossed brown and black and you got brown, that means that brown is going to be dominant. And if you crossed wingless with normal wings and you got normal wings, that means normal wings is dominant. Okay, and here in the box, it says all the things that we already know, and then that assume that the genes are not found on sex chromosomes and indicate the color alleles with the letter B and the wing alleles with the letter N. Okay, so A, the genotypes of the flies in the P generation are, so for the female, it's brown and wingless, and we know, since it's true breeding, that it should be homozygous. And we identified that brown should be dominant, so it's going to be homozygous dominant for the color, and then wingless. And remember, we said normal wings are going to be dominant. So the female would be homozygous recessive for wings. And then for the male, it's black, so it should be homozygous recessive for color and normal wings, so homozygous dominant. Okay. B, the genotype of the flies in the F1 generation is, so we know that it should be heterozygous if we cross 2 true breeding flies with different traits. And it was brown with normal wings, so it would be heterozygous for both traits. Okay. C, you now take an F1 female and cross her to a true breeding black wingless male. This male's genotype is, so again, it says true breeding, which means homozygous for the traits, and it's black and wingless, so they're both recessive traits, so it's going to be homozygous recessive for both of them. Okay. D, you count 1,600 offsprings in the F2 generation. If the wing and the color traits were linked and no recombination occurred, this is important, you would expect to count, and then you have these options. Okay, let's first look at the cross. So we crossed an F1 female to this one here. So this was our cross. Let me actually do it in blue. So we have a heterozygous female crossed with homozygous recessive male. So we're going to actually not do Punnett squares for this because it specifically said no recombination occurred. So we want to think of just the cases where there isn't a recombination. So let's think of this in terms of chromosomes. So for instance, for this one, it's obvious that the chromosome is going to look like this, right? You have 2 chromosomes that are identical, that both have the same genes, and they may or may not have different alleles, and in this case, they don't. And for this one, let's kind of trace it back to where it came from. It came from this crossed with this, right? So basically, it's this chromosome from the mom and this chromosome from the dad. So this one should look like this. Okay. Now, if we cross these and there is no recombination, what are the only genotypes that we can get? Let's find out. So what if we have this one and this one come together? It would be this chromosome and this chromosome. And then like this one and this one would basically be the same thing. And then let's look at this one with this one. It would look like this. And then this one and this one would look the same. So these two are the only genotypes that we can get if there is no recombination occurring. So this genotype is heterozygous for color and homozygous recessive for wing. And this is homozygous recessive for both. So this is brown and wingless, or oops, I'm sorry, I think I made a mistake here. It should be a capital N because this one here. Okay, so it's going to be N, capital N, lowercase n. So this is going to be black winged. Okay, so these are our only two options, meaning there's 50-50% chance, and out of 1600 that would be 800 of this and 800 of this. So if we go back to our list here, brown winged flies would be 0. Black winged is what we have here, so it'd be 800. Brown wingless is what we have here, so it'd be 800. And black wingless we don't have, so it's 0. And the genotype for brown winged would be, if we think about this cross here, the only genotype that would be possible would be this. And for black winged, for black winged it would be homozygous recessive for color, then heterozygous for wing. Brown wingless would be this, and black wingless would be this. Okay, so this is if no recombination occurred. Now let's go to E. When you count the F2 generation, you really get 85, 728, 7112, and 75 for each of these. So what is the genetic distance between the color and the wing genes? So let's first identify which ones are the recombination ones. So remember, these two were the ones that we saw in D where no recombination occurred. So that means these two are the ones with recombination. So remember how you calculate recombination frequency, RF? It's the number of recombination divided by total times 100 for percentage. So that would be 85 plus 75 divided by the total 1600 times 100. And if you calculate that, that would become 10%. So our genetic distance is 10%. Okay, let's now move on to F. A series of fruit fly mating shows that the recombination frequency between the genes for wing size and the gene for antenna length is 5%. List all possible recombination frequencies between the gene for color and the gene for antenna length. So we have 3 different genes that we're looking at here. The wings, the color, and the antenna, or the antenna length. Okay, so from E, we calculated that the genetic distance between wing and color is 10%. And in this problem, it tells us that the genetic distance or recombination frequency between wing and antenna length is 5%. So let's think of this on like a line of a chromosome. So since 10% is greater than 5%, we know that wing and color should be farther apart than wing and antenna. So let's put them on each end. So we have wing here, color here, and this here is 10% distance. So the other thing that we know is that wing and antenna are 5% apart. So antenna might go in the middle, where this is 5%. Or maybe antenna is on the other side, so it kind of looks like this. You have wing and color, 10%, and then the antenna is actually over here, and this is 5%. So these would be the two different types or possible recombination frequencies. I hope that was pretty clear. It might have been a little bit tricky, especially all of this, but let me know if you still can't understand, and then we can either do a call or something to help you better understand. And also let me know if you have any questions for question 2 and question 3. Okay?